No, I don't think so. I think Winfred
nice.
did not. I think Winfred, you have to unmute yourself.
I'm unmuted. Rudolf,
nice.
I can hear you well, but I did not hear the questions which were asked to you.
You have to choose the English channel.
I'm in the English channel. How can I select the English channel?
It's on the base menu. It's called English. Where is translator? It is where you find the chat function.
Oh. Okay. I've chosen it.
Now you go Bildschirm freigeben.
Hello, welcome everybody. I hope you can hear me.
Yes, very nicely.
Very good. A very cordial hello also from my side. It's really unfortunate that I could not travel to your beautiful country and to Sochi. I really would have loved to meet you all, but I'm very happy that we can meet now via Zoom. So hello everybody. So what I'm going to do today is actually I will explain you how we educate and do research in a PhD program, in an international PhD program on molecular, cellular and clinical allergology, which we, so Rudolf Valenta and myself, have built up over the last 10 years, so to say. So the international PhD program has to define topics which have a very high likelihood for success, of course. So we focused here on allergy research in different fields. It has to form a very strong group of participants from the side of faculty members, but certainly also from the recruited students. We then have to find structures and ways how to execute and run the program. And of course, we need constant evaluation of our own performance by third-party groups such as an international advisory board and peer review. Now, the major research areas we are focusing in our PhD program, in the international PhD program, is molecular allergology. And Rudolf Valenta has told you already lots about it in his recent talk, where we define new allergens, we find ways how to express allergens to formulate vaccines. We also study cellular allergology, where we study the cellular mechanisms in effector cells, in T cells, and in model systems. And then we also have a very strong clinical translational part in which we actually translate the findings from the molecular and cellular findings into clinical questions and trials. And this makes the program really strong and makes it also very interesting for the people working in the different fields, because they get a glimpse into related fields of their work, and they also see how their work, for instance, in basic research is developing in applied research. Now, these are just for the beginning a few numbers. I told you we are running the program since almost 10 years. We have combined 16 faculty members within the program. Up to today, we have recruited 68 PhD students into the program from 26 different home countries. So we really recruit on an international basis and based on excellence, as you can see here, in order to give and deliver the appropriate education to the students We have prepared more than 100 lectures and seminars and lecture series actually on courses for the students on the university level. There's also another very interesting part for the students built into the program, and these are so-called stays abroad, which offer the students the possibility to go actually during their research in the PhD program to interesting laboratories where they can actually complement their work, which they are doing here in Vienna. And these research stays abroad are very much appreciated. Now, certainly, the work which is being done is also submitted to peer review and then published. And up to today, we have published around 200 papers out of this PhD program. So it is really, there's a constant flow of new knowledge and research coming out of this. And more than 30 students have already finished their studies and are now doctors and PhDs. So we are very proud of them. And they have found now jobs in university setting, in the academic setting, but also in industry. Now, how is the PhD program from the beginning? How was it positioned initially within our university? So you can see here this nice house, our dacha, so to say. It has a very strong roof built by the Medical University of Vienna. And you can see here the original faculty members who actually worked in the different fields and helped to recruit students. The whole was built up on research grants, on large research grants, which were either granted to Rudolf Valenta or other members within the faculty. And what we did actually, we combined these people here around different basic scientific aspects and medical specialties. So we combined actually molecular biology, protein chemistry, structural biology, cell biology, and animal models together with medical specialties from dermatology to pediatric diseases, from respiratory diseases to vaccinology, as you can see here. And this made the program from the beginning very strong and very attractive, not only for the PhD students, but also for us, because it was really big fun to build that up and to keep it running. So what we tried to build up is actually a cradle for the new generation where we can actually pass our knowledge to the new generation so that the knowledge transfer keeps running and keeps science going and moving. Now, in detail, I'm going now a little bit more into the detail how much actually different steps are behind such a program. And you can see here the different steps in which the program was running. So it all started in 2012, where we filed the first application to our National Granting Society, FWF. We had then a first hearing, as you can see here. It was successful and approved, and then we could recruit actually in the year 2013, and we could actually put out a call for the first recruitments. We recruited then the first class of students, around 20 students, which we called the class of 2013. During this first application, we received enough funds to have another call a year later, which formed then the class of 2014. Now, the whole process was evaluated 4 years afterwards, in the year 2016. We were successful within the first 4 years, and we got a very high rating. So we could then, in the next step, hire the next class, the class of 2017, as you can see here. And then the program did run until 2020. Now, we did not fall idle after 2020, because that would have been the end of the program. We thought, well, the program is running so well, and Rudolf Valenta, all of those who know him, he was not sitting in his office and hoping that some new grants would come in and fall into his lap. He very actively worked on getting even better funding and more funding, and he succeeded in actually running a so-called then third application, if you wish, where he built up the Danube ARC, the Danube Allergy Research Cluster, and now our PhD program could become part of this Danube ARC. Now, the special thing of the Danube ARC, and we will see it in the next slide immediately, is now that we enlarged our community and we reached out from the Vienna region, from the city of Vienna, to the lands around Vienna, to Lower Austria, which for us is the Danube region, but we know that the Danube region is reaching much further, also to your parts actually. And we could actually include new groups into the Danube Arc, and we could actually have new course for students, as you can see here, and the class of 2020 was only hired last year, and these are the students, most of the students who are now with us and doing research with us. Now, we are already thinking about the future, and there are movements ongoing for the establishment of a so-called excellence cluster. And we will see how this is running, because we want to continue with our program beyond the year of 2023, because it's so successful and of high caliber. Now, here you can see the faces around the faculty members, the students from the class of 2013, students of the class of 2014. These are the students of the class of 2017, very international, very gender balanced, as you can see here. And this is now the new class of 2020, students from all over the world. You will see it immediately, the details. So what we have built up now is the Danube Allergy Research Cluster with the embedded PhD program. The groups have changed a little bit here. There are different names, new groups. Others have left the program. So this is also important for us to get in so-called fresh blood into the groups and fresh ideas. And as you can see here now, the umbrella is a little bit different. So it's now not the medical university on its own, but it's the Danube Allergy Research Cluster, where we get money from the state of Lower Austria as funding. And it is now built on different universities, not only from Vienna, as you can see here, but also from Lower Austria, such as the Karl Landsteiner University in Krems and the Austrian Institute of Technology in Tulln. Now, let me go a little bit into the details, how the program is running. So this is the flow chart of the different bits and pieces, how actually the processes run in the program. And the red arrow here, this should demonstrate how a student, a PhD student, is actually moving through the program. So we get applications at the beginning to our course. Then we have very stringent hearings and evaluation schemes. If these are passed, the students will be admitted. They will do research and training. They will do and submit and provide the publications. And then in the end, they will finish with a thesis, with a defensio. And some of them will stay with us. Others will go to other universities. And the third ones will go to the industry and will have very attractive job offers. Now, in order to run that main process here, we of course need lots of processes which actually help us to run the overall program. And this is a steering committee, as you can see here, admission committee, where the faculty members help together actually and run the different groups. We also have students committee who actually interacts with the speaker and with the deputy speaker in order to get ideas and input from the students. For each student, a thesis committee is actually built up not only consisting of the faculty supervisor, but also of one intramural, so from the same university, and one extramural scientist, in order to have a very good supervision culture for the student. The idea being behind that is that the students should not be always completely alone with his or her supervisor, but should also get advice from mentors outside. Now, of course, there are rules and regulations which we have to fulfill, which feed into these processes. And then here on the bottom, these are small inputs, as you may think, but they are very important. These are all these support processes from human resource management, from the rectorate of our university, from the funding sources, and so on, which feed into the main process. So this is running very well in our hands, and we are very happy about this stringent structure, which helps us here. Now, how does the student education for the individual student look like? And this is depicted here, for instance, for a student who is doing his research and started his research in 2020. So you see here, the student filed an application. We did the eligibility check initially, and then we invited the student for an interview and for a presentation of the scientific work and also of the scientific work of others in the form of a journal club. And then if the whole evaluation was running well, and if a match was found also with a faculty member, then the student was admitted to the program and started his research like in October last year. Now, thesis committee was established very early on when the thesis was formulated and the topic of the thesis and the milestones were defined. And this had to be presented actually in form of the thesis committee, which we usually do in form of PhD symposia and or retreats. And this is following on year by year, as you can see here. So we have a kind of a regular process here. Some students go to a stay abroad. They do research also outside of the laboratory of the faculty member. And then within 3 years, most of the students have enough scientific material and scientific data collected so that they can write first papers, first reviews, and then they can start to think about their defense and their doctoral thesis. Now, this is just these are just examples where our students go for stays abroad, for instance. So they do not stay idle in Vienna. Some of them go to Harvard, others go to the Karolinska Institute, and so on. And this is also very important for the building up the connections and the networks for the later jobs the students are seeking. So they get first job offers, then also already for the postdoctoral career. And we are very happy about that. And that also brings in and brings very close to us scientists from other faculties and other countries. All our endeavors, certainly the great research which is done in Vienna, but also the doctoral program which is contributing to that research, has actually positioned Vienna and the Medical University of Vienna in the worldwide ranking for allergy research on a very prominent 7th place, as you can see here. And we are very proud of that because there are only a few competitors in front of us and many actually behind us. And this is certainly continuous work of Rudolf Valenta and his disciples in the field of allergy research, and also our long-term interest in the field, which is very important. Now, this was the part which I was planning to tell you about, the Danube ARC, the Danube Allergy Research Cluster MCCA PhD program. And now I would like to show you a few examples, actually 2 examples from my laboratory, which model systems we are able to build up, because I realized that you guys are interested actually in translational models, in genomic data. And I would like to share with you 2 ideas and 2 research fields which are certainly connected to allergy research, but which have maybe some interesting aspects for you in there. So you may have heard already from Rudolf Valenta that actually allergy has many phases. There are acute allergic manifestations like rhinitis, conjunctivitis, and urticaria, which are very nasty to the patient actually. There's also chronic manifestations of allergies such as asthma affecting the lungs, but also the skin, such as atopic dermatitis. And atopic dermatitis can affect small children, as you can see here, but also adults. And allergy can be very dangerous because it can lead to life-threatening events such as angioedema, where actually your mucous membranes start to swell and then you get problems to breathe. And we would like to avoid that, and we would like to understand the disease better, how it is initiated and how it can be treated. And one of the problems, ladies and gentlemen, is actually that the prevalence of allergic diseases is growing continuously. And this is not only true for the milder manifestations, hay fever, and also eczema. This is also very true for asthma. And this poses a big problem to society. And we would like to understand, together with Rudolf and our colleagues, what actually the primary factors in allergic sensitizations would be. Are these mostly genetic, endogenous host factors built into the patient already, where you cannot maybe much do, or are there also exogenous factors which contribute to the disease and which might be amenable to changes and protection? So the questions we are asking is, why do certain individuals become sensitized towards certain allergens, and why are they developing allergies? And what might be the primary factors? And here comes Alina Neunkirchner. She was one of my PhD students. She started a PhD already before we have established the MCCA PhD program, but then she helped us to structure the program very well as a postdoc in the program. And what she did is actually, she was very much interested in mugwort allergy. So you can see here, mugwort is a weed which is growing during summer. And we realized that you guys in the Russian Federation have lots of mugwort, and we have seen whole fields of mugwort actually towards Vladivostok, and you may also have it in the Moscow region and maybe also in Sochi if you look around. And mugwort is a very bad weed because it flowers during summer, late summer. It is starting now maybe, and is a very strong sensitizer. And she wanted to study mugwort allergy in detail, and she actually created a mugwort-specific mouse model where she took the genes from human T-cell receptors, human T-cell receptor genes, and introduced them in the mouse. And she also introduced the HLA molecules from human beings into the mouse in order to create double transgenic mugwort allergy mice. And these mugwort allergy mice, when they were receiving mugwort aerosol, mugwort pollen extract, as you can see here, they reacted very strongly with their lungs. So their lungs reacted very strongly and they developed a very strong PNH, as you can see here, and airway restriction. And this was not the case with those mice which did not have the specific T cells and who did not have the specific HLA-DR molecules, as you can see here. And also the wild-type mice with this sensitization protocol did not develop, if you wish, mouse asthma. And you can see here also the development of mouse asthma of airway hypersensitivity measured invasively. So there was really a very strong phenotype, and you can see the very strong phenotype also here in histology. So this is lung tissue. This is healthy lung tissue of wild-type mice which actually were breathing mugwort pollen extract, so they did not get asthma. The bronchi did look perfectly well. The same was true for the HLA, DR1 mice, no problem at all. And also for the T-cell receptor transgenic mice, no problem at all. But if we combined the T-cell receptors together with the human restriction elements and the presentation molecules, the immune response genes, then we got massive mouse asthma. You can see here the pathological bronchi, lots of infiltration actually here, paravascular and peribronchial infiltration, so a very strong disease phenotype. And now we wondered if we would now increase numbers of T regulatory cells in such a system by using a special trick, actually, by actually increasing the levels of interleukin-2 in the system. We wondered whether that would ameliorate actually disease and mouse asthma. You can see here how well this treatment works actually. When you administer interleukin-2 complex with anti-interleukin-2 antibodies, then you can actually increase regulatory T cell numbers by 10-fold. And this 10-fold increase of regulatory cell numbers actually helps to ameliorate disease, as you can see here. So if you give those complexes, then the mice cannot be sensitized anymore, indicating that T regulatory cells seem to protect the mice from becoming sensitized. Now, this was very interesting and good news. And in the next step, we wondered whether this can also be done prophylactically. And this is the prophylactical scheme which we applied. And you can see here that when we started sensitization and then came, and you can see here the sensitization is already starting after the second exposure here. This is the third exposure. IgE levels are increasing constantly, while this does not happen in the group where we increased T regulatory cell numbers with the help of interleukin-2. So what we show with this is actually that T cells are important for initiating the disease. You need the T cells which interact with HLA-DR1 in order to get sensitized. But if you regulate the T cells, you can actually put a stop sign and a stop onto the activity, onto the allergen-specific activity. Now, in the last part, in the last few minutes, I would like to show you data from Peter Tauber in the lab. And he was now picking up this idea of interleukin-2, that interleukin-2 is crucial for the development and maintenance of regulatory T cells, because mice deficient in interleukin-2 develop various autoimmune diseases. Disrupting interleukin-2 results in uncontrolled lymphocyte proliferation. IL-2 upregulates FOXP3 expression in human T cells, and we have shown this for the mouse T cells, and the expansion of regulatory T cells with interleukin-2 and anti-interleukin-2 antibodies can be beneficial in different diseases, and among them also allergies, as we have shown in our allergen-specific model. And he now wondered whether we always need these interleukin-2, anti-interleukin-2 complexes, or whether we could also do that with certain drug treatment with small molecular inhibitors. And he wondered whether we can selectively increase interleukin-2 secretion by T cells by adding small molecule inhibitors of T cell activation, actually. And what he found is actually a compound which is a PDK1 inhibitor, BX795, which selectively, as you can see here, increased interleukin-2 production when the cells, and in that case these are Jurkat T cells, receive a signal via the T cell receptor. Other substances which are very well known to you, like cyclosporin A or rapamycin, did not do the job. So it was very specific for this compound. And then we wondered, well, what is this compound doing to the cells? And he actually differentiated now cells, T cells, from our double transgenic mice in the presence of different stimuli, in the presence of allergen, as you can see here, in the presence of allergen and certain cytokines, which would actually induce regulatory cells, and also in the presence of the allergen and the compound. And we were asking, well, what would be the phenotype now of these cells, which are overproducing IL-2, and we call these cells TH-IL-2s because these are T helper cells producing interleukin-2. And much to our surprise, as you can see here in this principal component analysis, which we performed after sequencing genes produced in this so genes upregulated in the T cells after stimulation. So we did mRNA sequencing here, and the mRNA sequencing data showed that the naive cells did build a certain cluster here. So these are 4 different experiments. They cluster very nicely in this principal component analysis. The effector cells also co-cluster together, so they form a group. And here in red comes the ITregs. They form a third cluster, if you wish, and very close together with the ITregs cluster, the TH-IL-2 cells. So we realized, oh, very interesting. We do not need TGF-beta and anti-interleukin-4 and anti-interferon gamma in order to create ITregs. We can do it in the presence of a T-cell receptor stimulus and this BDK1 inhibitor, BX795, and generate this cell phenotype. Now, these are the summary data of the different genes we have found to be expressed, overexpressed, or repressed, and you can see here again the nice co-clustering between ITregs and TH-IL-2 cells, while T effector cells and the naive cells, they were completely separated from these other 2 groups. So indicating that this drug, this small molecule inhibitor, is actually inducing something like an ITreg, T regulatory cell development. And now let's come back to allergy. The very interesting thing, apart from the fact that these cells overproduce interleukin-2, is now the fact that they stop to produce type 2 cytokines. So they do not produce interleukin-4 like the effector cells. They do not produce interleukin-5 like the effector cells. And they also do not produce IL-13 like the effector cells. And very interestingly, and this is very much in contrast to classical ITregs, they do not express FOXP3. So what we have defined here, although they are highly activated, as you can see here by CD44 expression. So what we have found here is actually a new T helper cell type, which is very, very similar to ITregs according to RNA-seq data, but which is in certain aspects different from ITregs because, for instance, the signature transcription factor FOXP3 is not expressed. Now, in mice, so, and this is now the translational process of the whole endeavor, is in mice, PX dramatically and significantly reduced Th2 inflammation. And this can be seen here by reduction of GATA3 expression, so GATA3 being the signature transcription factor of Th2 cytokines. It reduced interleukin-4 production, as you can see here, significantly, and also interleukin-13 production in mice which were challenged, now again with mugwort pollen extract, and as a result of that, also eosinophilic influx into the lungs of these mice was significantly reduced. And this, ladies and gentlemen, brings me already to an end. What I've shown you today is actually that we have identified a new substance which promotes IL-2 production and the secretion of certain cytokines in allergen-specific T cells. They polarize naive T cells towards a distinct phenotype, as you can see here, TH-IL-2 cells. These cells secrete increased levels of interleukin-2, and the interleukin-2 I did not show you is biologically active because it is important for the maintenance or upregulation of certain markers of T regulatory cells and Bx suppresses Th2 inflammation in vivo. And this should have been only 2 examples of how we try to translate from the mouse cages to the patients, from the patients to the mouse cages, allergy research in Vienna in a PhD program, which we have built up over a decade now, and which we are planning to promote and run hopefully for another decade. And we invite you all and your students to apply during the next call. So hopefully this will be in 1 or 2 years, where we can open another call for the program. In the meantime, there's always places in the one or other laboratory for your students who would like to come to Vienna and who would like to apply. And with this, I say thank you very much for the kind invitation. Once again, I would have loved to join you guys in Sochi. I know how nice Sochi and how nice Russia can be. And I really regret that this time it was not possible. The more I'm happy that I can be with you in this format. Thank you very much. And many thanks for your attention. And I'm happy to take your questions.
Thank you very much. So any comments or questions, or I, as always, can start with? So about this graduate course, it looks like it works very well for the given university, specifically Medical University of Vienna. And is it kind of a product of systematic whole university approach, or it's an initiative of a given group of researchers and the professors? So are there any other groups in genomics or in other fields of studies, or it's just a successful example of allergology team work?
Yeah, this is a very good question. Thank you for the question. So actually the initiative came actually from our funding organization, from the national funding organization FWF, who actually started with this form of programs like 10 years ago. And we, as faculty members from different universities, had the ability to apply for such grants. Of course, these applications were highly competitive and they had to be focused on a very clear idea. And yes, there are other doctoral programs which are run in a similar format. However, I have to say, we are most probably the first PhD program who found additional funding after the official end of the funding of the granting of the original granting organization. And this was only possible because allergy research is so strong in Austria and is actually also, kind of going to different parts, also to your places. And because we were able to seek for additional funding.
Thank you very much. Any other comments? Or I have a second question, actually. So Russia these days face a question how to prevent so-called brain flu, or tries to keep young scientists in Russia. So how do you feel in Austria? So is there any such kind of question or problem, or a country instead stimulate this academic mobility across the countries, or specifically European Union and so on? So how it's going? Is it any problem? So it's a specific program are targeted to keep scientists in Austria or vice versa. So how it's going?
Well, this is a very good question. And I can give you 2 or 3 answers to that question. So question number 1, our generation, like Rudi and myself, we had to go abroad in order to promote our scientific careers. So, and for us it was pretty clear to go to the Americas, to Canada, to the US. However, we had always the possibility to come back to our mother university. So it was not forever, but it was like an interim stay in another country with a very strong interest in research. Now, when I think about how to keep people in your country, in Austria, in Russia, then I can only say that programs like the PhD program we are running, they are a perfect ingredient, because I can tell you when we build up the first, and we had the first course, then other students who did not apply for our program, but who were PhD students just in other labs, they got very interested in actually what is going on and why is there a group formed here and why is there a class of MCCA 2013 and then there is a class of 2014, and they wanted to become part of that. Yeah? So because it was so interesting, because we have of course also additional activities like retreats, symposia, we bring in scientists from the outside. We have seminars, international seminars and so on, from scientists from all over the place, also from Russia, who like to come to Vienna, give seminars, give lectures and so on. And this is, raises the attractiveness for students to stay actually in their country, right. And to consider a very solid and high level education in the country.
Thank you very much. And if there are no more comments or questions, let me thank you again for your excellent talk and close this part.
Thank you very much.
Thank you.